So guys, in class, we talked about transcription, right? We talked about how we went from DNA to RNA and how that physically happened. We also have already talked about how DNA makes copies of itself. But now that we have this piece of RNA, what do we do with it, right? How does it turn into a protein? And that process is actually called translation. Now, protein synthesis is transcription and translation put together. So we're going to talk about translation now. So this is the process by which the ribosome takes that coded messenger RNA and decodes it, right? And decodes it, turns it from the language of DNA and RNA into the language of a protein. And then eventually that will make that functional protein that is part of your body. If you remember, amino acids will fold into complex 3D structures. You remember primary, secondary, tertiary, quaternary, right? All of that was really determined by their R groups. So if you remember, here's an amino acid. This picture is an amino acid from Unit 1. We have an amine group and a carboxyl group, right? The amine will bond to the next carboxyl. The hydrogen sticks off here. Everybody has one. But this R chain, this side chain, has different chemical properties. And since there's 20 different amino acids, each one of them has their own protein properties. This is really small. I don't expect you to be able to read it, but if you remember, we had one where we had acids, we had bases, we had ions, we had, side chains that were all carbon hydrogen chains, and they were hydrophobic and some were hydrophilic. And so all of those different properties come together to make that functional 3D shape. And if you remember, we talked about if you change the shape of a protein, you change its function. And we don't want to change the function of our proteins. We want them to work properly. So how do we actually translate? Okay. So in transcription, we only explained how you made DNA into one type of RNA. We made it into messenger RNA. And we talked about the processing of it and how you broke off little chunks and you took out the introns and all of that. Well, there's actually three types of RNA. There's messenger RNA, which is that long single strand of RNA nucleotides, and it's actually the code, right, from the bases of the DNA. It contains those codons, right, the codons. And each codon will be translated in this section into one amino acid. tRNA, transfer RNA, is another piece of RNA that starts out single-stranded, but then it folds up on itself to make this like clover-like structure. And the clover-like structure has a spot on the bottom where three bases are exposed. Those are called the anticodon. They match the codon. So we know which tRNA we need in order to match the messenger. And then at the top, there's a place for them to hold a specific amino acid. So transfer RNA's job is to take, it's to take the amino acids and bring them to the site of the ribosome. And then finally, we have to have the ribosome itself. Now, the ribosome is made up of two things. It's made up of protein and ribosomal RNA. So our RNA and proteins come together to make the physical structure of the ribosome itself. And that's where all of this is going to happen. So in messenger RNA, you have this big long strand that can be decoded and made into a protein. That's kind of like a silly video, but you get the idea. Then in transfer RNA, at the bottom, you'll see it has these three little bases, and it knows how to bond to the correct messenger RNA, which is the big long pink strand in this picture. It's carrying an amino acid, which then bonds in this long chain. And so, there's a new tRNA, it connects to the other ones, and so on. And the tRNA keeps joining. What's the big purple thing? Right, that's the ribosome. And so this is all happening in the ribosome. The messenger RNA gets together with the transfer RNA, and there's the proteins. Our RNA is the ribosome itself, and it goes down the messenger RNA, as you can kind of see in this little video. And what's coming out the top that looks like kind of like a scorpion tail is actually little amino acids bonded to each other. They're the little proteins. Eventually, the ribosome falls apart, and there you've got your RNA and your protein all put together. So you can kind of see the proteins up here. So how does this happen, right? What are all the steps? What's going to go on? So again, we're going to have an initiation, elongation, and termination, right? Initiation, we're going to start. Elongation, we're going to make things get longer, and then termination. So initiation is the start of it, and that's where you take the two pieces of the ribosome and put them together. And when we put them together, we add in a start codon here. So if you look, The blue thing here is a transfer RNA that is carrying the proper start codon. In the picture, they've got it kind of blown up here. You can kind of see in my cursors. AUG is on the messenger RNA. That's the codon. UAC is on the transfer RNA. That's the anticodon. It knows that only that proper anticodon can fit to start with methionine. So here we've got our start codon. Now we add some energy. GTP and ATP are used for energy. And now we've got this whole subunit put together. Okay. The tRNA carrying the matching anticodon attaches in the middle of the ribosome. Do you see how it's in the middle there? And that's going to begin this whole polypeptide chain, the whole thing. The ribosome will eventually move on the RNA and it'll start adding in more. Okay. So that's the elongation process. Inside the ribosome, there are 3 messenger RNAs in one codon, and it reads it one section at a time. So let's look at this picture, okay? So you see the 3 places we can put transfer RNAs. A new one comes in. Here it comes. You transfer the polypeptide to it, and the old one gets kicked out. So watch this old one. It's done, and it goes away. It's naked. It doesn't have an amino acid anymore, okay? So the ribosome has a place for 3 tRNAs to fit. 1 at the beginning is the attachment site. 1 holds the growing polypeptide in the middle. And then 1 is that exit point where the final empty tRNA will fall away. The ribosome is going to keep moving down the messenger RNA and making sure it properly aligns those anticodon-codon pairs until we get everything done. Then the ribosome moves the amino acids together, makes a big long chain. How does it know when to stop, right? Because the RNA piece is actually long. If you remember, it has a cap and a tail. Does it decode the tail? Does it decode what? And so it knows when to stop when it reaches one of the 3 stop codons. There are 3 of the 64 possible codons. There are 3 that don't match to any amino acid, okay? They don't match to a tRNA that's carrying anything. They code for something called a release factor. So if you watch here, we'll get this guy started back to the beginning. Here we've got 2 amino acid transfer. Okay, so that one's exiting. It shifts, leaving open an attachment place. Then this pair, the UGA, is the release factor. The release factor causes everything to fall apart because it doesn't have a protein, it doesn't have an amino acid, so everything falls apart and it just starts over. Once these release factors are in the ribosome, right, they could trigger everything to fall apart. So the ribosome, messenger RNA, the transfer RNAs, they all get used again. The messenger RNA is actually probably decoding with many polypeptide ribosomes on it all at one time. So this is not being wasted, it's just being reused. And so when we reach those stop codons, we stop. If you remember, in a nonsense mutation, that's when you end up with a stop codon in the middle of your protein where you don't want it and everything falls apart. So no matter how much messenger RNA you have left, it doesn't work because the release factors have caused it to stop. So in eukaryotes, translation happens in the cytoplasm as well as in the ribosomes, okay, on the rough ER. The process of transcription and translations occur in different locations in eukaryotes. Where does transcription happen? That's right, it happens in the nucleus, and translation takes place in the cytoplasm. That's correct. So in prokaryotes, translation can be happening in the same time as transcription because they both happen in the cytoplasm, right? They're just do. So you can be making the RNA and all of a sudden you can be decoding it at the same time. So if you look at the picture, since there's no nucleus, the DNA is this big circle, mRNA is being made, it's kind of this green molecule, and the polyribosomes, these 3 different ribosomes here, are making proteins all along. Up here in the eukaryote, the DNA is in the nucleus, it makes RNA, we process it, then the RNA comes out of the nucleus and then it gets decoded, right? Then you have translation. So what is the code? Like, how do we decode this? Some scientists really worked hard and they were able to discover the actual letters, AUG, CUG, whatever, and what they code for. The genetic code is universal to almost every single living thing on the planet. Like 99.9% of all the organisms have the exact same genetic code with no differences. That tells me that evolutionarily speaking, the genetic code evolved really early in the process. So the same three-letter codes can be read exactly the same way in all those living exceptions, except for a few bacteria, things that live in like hot springs or deep in the ocean. Since all the genes coded from the same language, we can insert genes from one species into another, and we can actually read them. That's genetic engineering. We'll talk a little bit more about that later in the unit. Bacteria and eukaryotes are a bit different in how we process our messenger RNA. If you remember, eukaryotes have the nucleus, so we have to do some things. Bacteria don't do that. They don't have that. So if we want to have a bacteria code for a human gene or a plant gene, we actually have to go ahead and process the RNA, put a cap on it, a tail on it, and get it all together so it's ready to go. And so all genetic engineering really takes advantage of this universality. Currently, scientists will use both bacteria and yeast, typically, because they're single-celled eukaryotes, to help generate large quantities of proteins, things like insulin, etc., things that we're going to use in large quantities. So I want you to be able to read the code. You will never have to memorize the code. You just have to be able to read it. Okay? So when you start, you always start with the start codon, which is AUG, and every word is 3 bases long. So here is a chart. I'll give you one for your notebook. Here is a chart that you can read, okay? So the first base is over here on the left. So we're looking for a letter. It's over here on the left. We start there. Then we go to the top for the second letter, right? And then we go over to the left and we find the third letter. And so the middle base is on the top, the third base is on the right, and then so on, okay? So I want you guys to decode this one, all right? And we're going to do this in our Edpuzzle. So I want you to find AUG. That's methionine, MET. Now I want you to find CUG, leucine. Okay, keep going. And so when you finish each one, it's pretty easy. And so we're going to practice these. I'll have you write these in your notebook, and then we'll go through them in Edpuzzle as well. All right. Now, the one on the left is the common way it looks, right? It's usually in a chart, but it can also be this circular thing. So again, find AUG. So you start in the middle and you work your way out. AUG, methionine, right? I kind of like the circle chart. To me, it's a little bit easier to read, but it's not the one I typically see on the AP test. So I'm going to go ahead and stop us here. We'll practice these. You guys are going to have a worksheet that'll go over how to do the genetic decoding and all of that. But that's probably one of the simpler things you'll do in this class. Thanks, guys.